Howdy! Today we're going to talk about transport across the membrane, which is the second part of the cell membrane. The first part was just the types of proteins and things that you can find in the cell membrane. Now we're going to see how those things actually go to work in terms of transport. So I made this little outline for you to kind of help you visualize the different types of transport. There's two main kinds. There's passive and active. So those are the two big categories. Passive requires no energy whatsoever. So it's just passively happens. Active does require energy. Okay? So that's the two main categories of transport going on inside of your body as we speak. Under passive, there's three different types: simple diffusion, facilitated diffusion, and osmosis. And active transport, there's two types: endocytosis and exocytosis. But under the category of endocytosis, depending on what they're taking into their bodies, there are two types: pinocytosis and phagocytosis. And so those two guys right there are just subcategories of endocytosis. So we're going to start with passive, simple diffusion. All right, here we go. Weehaw. Passive transport is the movement of molecules without the use of energy. So this happens naturally. There's absolutely nothing you need to do to make this happen. It's kind of like when you're making Easter eggs and you get the cup and you put the water in and you put a drop of food coloring in it. If you just leave it alone and you come back to it later, the whole entire thing is going to be completely filled up with food coloring. The food coloring is not going to stay in a little bitty dot in the middle. Why? Because of the diffusion of molecules between the water and the food coloring is going to cause them to randomly spread out. And that's exactly what happens during diffusion. In simple diffusion, things like water, oxygen, and other molecules move from areas of high concentration to low concentration down a concentration gradient. So what that means is kind of like an elevator. If you have a nice elevator packed full of people, once you open the doors, well, then people are going to come out and spread out till you have even numbers on both sides. Yeah, I know people usually want to get off the elevator, but if some of them had to stay in there, they would do so and just kind of make it so there's half in here and half in there. So if we look at this picture down below, we can see that the cell membrane, the lipid bilayer, is allowing these molecules to slide right through them to the other side. And they'll keep on going until they have equal numbers on both sides. Now, it doesn't mean that they're going to, once they get there, they stay. They can migrate back over here, but for every one of those, another one's going to migrate over here. If two migrate over here, well, two more will migrate over here. So there's still movement of particles, but they do it at an even rate, so that way they don't get reconcentrated again. So this could be something like glucose. So when you eat lunch and here's all your cells, pretend that's a cell, and here's your lunch particles, your glucose. So if there's too much in your bloodstream, then they're going to start to diffuse into your cell until you have equal numbers of glucose in your bloodstream and in your cells, and that'll make your cells happy. So simple diffusion, just movement of water, oxygen, and other small molecules from high to low. Okay, then we have facilitated diffusion, which is, we looked at one of these little proteins the other. They are assisted by channel or carrier proteins. Now the word facilitated means to help. And so this is just plain old diffusion with help. That help comes from a particular type of protein. So I still have a high concentration of particles in one place, in this case outside of the cell, and a low concentration inside of the cell. And so all these molecules want to get through, but the only problem is they're too big to just go through naturally by themselves. So they actually have to get help and go through either a channel or an ion protein in order to get through to the other side. And they'll do so until we have equal numbers both inside and outside of the cell. Third type is osmosis, which is just simply the diffusion of water. So diffusion, we usually refer to other particles, but if we're talking specifically water, then we call it osmosis. And what's really interesting about this one is that, well, here, let me show you on the picture. Okay, so over here, I've got a beaker, and it's got this membrane right down the middle. That's this dotted line right here. That allows small things to go through, but not big things. So the blue things are small. That's my water. The big things are sugar. Okay, the sugar is too big to bounce through to the other side. Because normally what would happen in simple diffusion, I've got so many of these sugars over here and only a couple over here, well, they're going to want to go over to this side, but they can't. They're stuck behind this wall. So what do you do if they're stuck behind the wall? Let me get a different color, not the red or the blue here. Well, if they're stuck behind there, then the water says, hey, I got too many of us on this side, not enough on that side. Hey, let's go over there. And so all the water molecules go over there until there are equal number of water molecules on one side and the other. Now, even though the glucose, the red, stay put, what we see now, look what happens to the difference in the water levels. They were the same before, but now it's higher on one side than the other. Why? Because we want the concentration of water to be the same on both sides. And since I can't move the glucose over, the water says, no problem, I'll scoot over for you. And so now we have the relative same glucose per water ratio as this side. So if we count up however many glucoses and water on both sides, we'll find out that the ratio is about the same. And so there's something kind of interesting with osmosis is that we see whoever has the more solute, I can't spell, and my pen sucks. Whoever side which has more of the solute is going to suck. Solutes suck. So I have more solute, okay, glucose on this side. They are going to suck the water over to that side right there. And then that's what changes the water level from here to here, is because it's sucking all the water over to that side. So when we think osmosis, stupid light, excuse me. Trying to get the light back on. Come on, turn on. I am here. I really hate this light thing. Okay, my light doesn't know I'm here, so I'm just going to keep going in the dark. I don't care. So anyway, so solutes suck water towards them. That's why we need a whole bunch of salty potato chips. You have so much salt in your bloodstream that it sucks the water out of your cells, and that's why you get really, really thirsty. Okay. How osmosis affects cells. Now, if I take a cell, so I have a beaker of something, of some solution, and say I put a red blood cell. Oh, here, I'm sorry. I'll make that red for you. How's that? Okay. If I put this red blood cell into some water, depending on how the water is around the outside is going to change what happens to the cell. If the cell is placed into an isotonic solution, what that means is however many particles of stuff, salt, glucose, whatever, are on the outside of the cell, that will equal the concentration of particles inside the cell. That is a happy cell, because that is a normal, everyday functioning cell. So when the concentration of stuff outside is the same as inside, we get a happy cell. Now hypotonic, hypo means under. And so what happens is, let me get my beaker, water, blood cell. If there's a higher concentration of solutes inside the cell, so I got a whole bunch of particles in there and only a couple under, lower, so that's where the hypo comes from. Hypo means there's not a whole lot outside the cell. So what's going to happen is that my cell is going to see all this water, because there's more water out here than inside, and it's going to suck the water in, osmosis. What happens to a cell when it sucks up a whole bunch of water? It explodes. We call that cytolysis, or cyto, cell, lysis, to split. It explodes. Well, what if I do it the other way around? In hypertonic, hyper means more. And so if I take my beaker and my water and I put my cell in there, and now I got tons of particles in here. Say I sprinkle salt in there, and I only got a couple in there. Well, what's going to happen? There's more water inside than outside the cell. So water's going to go where it's needed. He's going to leave the cell, causing the cell to shrivel up and die, like a little raisin. That is called plasmolysis. So we have cytolysis and plasmolysis. This one explodes, this one shrivels up like a little bitty raisin. So let me show you what some real cells look like, or at least some drawings of real cells. So here's my happy solution. Okay, I got the same amount of water going in as the same amount of water going out. My red blood cell, an animal cell, is very happy. Here's a plant cell. Same amount of water going in, same amount of water going out. Happy cells. Happy, happy cells. Hypotonic, low amounts of stuff outside, way more inside. So what's happened? Water rushes in to the point like a water balloon, it explodes. But this plant cell has this big thick cell wall around it that's actually going to protect it and just cause it to swell up. Now it can break, but usually it'll just swell up. Now in hypertonic, hyper, so the solution has way more stuff than it should, so there's tons of stuff out here, only a couple in here. The water is going to get sucked out of the cell, cause it to shrivel up like a little raisin. In a plant cell, what happens is that the cell membrane kind of gets sucked in. Parts of it stay stuck to the cell wall, but the cell membrane is actually getting sucked in to the middle, and so it's losing volume, it's losing water inside. Again, the cell wall isn't going to shrink, it's going to collapse in on itself a little bit, but it's the cell membrane where the most damage is going to be seen. Okay? So, hypo, under, which means not a lot of junk in the water around it. Iso, equal, hyper, way more junk, solutes floating around in the water than inside of the cell. Okay, active transport. So now we have the second category. So all those, no energy. So now we have the second category, which is active transport, which means we need energy, usually in the form of ATP, adenosine triphosphate. So active transport is I need to move molecules into my cell, but they're way too big. They can't just go straight through a protein. How am I going to get them in? Well, first way is through endocytosis. Endo sounds like the word in. Makes sense. So endocyto, into the cell. This is a process of taking substances into the cell. So here's my little guy right here. This is my cell. Here's my food particle. He's too big to slide through, so my cell needs to eat him. So we know that the phospholipid membrane is double layered, so they're showing that with the red on the outside and the black on the inside. As the particle comes closer, we form a little indentation in the cell membrane, and it starts to engulf or wrap around it. And as you can see right here, the red is starting to come closer to each other, and eventually what happens is when it pinches off, since the outer layer is what wrapped around the food particle, you get an inside-out bubble. They're calling it a vacuole. We also call those vesicles too. And so what we have is an inside-out. So the inside of the cell is still touching the inside plasma membrane. And then this little space inside of here is made with the outside of the cell membrane like that. And so that's what we call endocytosis. Now, if that food particle is solid, we call that cell eating or phagocytosis. Phage means to eat. So this word right here means to eat, cell eating. But if it's a liquid thing, then we call it pino. Pino means to drink. And so that's cell drinking. So depending on what this particle is, if it's food, we call it phagocytosis or solid. If it's liquid, we call it pinocytosis. I just like saying it that way. You don't have to say it that way. Pinocytosis. Okay, but then it sounds like you're saying the word penis, and that's just no fun. Okay, so pinocytosis is cell drinking. Both of these fall under the category of endocytosis, which you can use. If you can't remember either of these two, just remember endo. Endocytosis goes inside of the cell. So take a wild guess as to what exocytosis does. It's backwards. It's the same exact thing backwards. So I want to get rid of something. So I want to get rid of my waste particles, or I want to spit out a protein. So what do? So I've got, here's my outer layer, which was the inner layer in here. Okay, inner layer. And it joins with the outer layer like that, and then look, it just becomes the outer layer again. And then what's the outer layer of the vesicle is really the inner layer of the cell membrane. And so what happens there is that, hey look, it just becomes part of the inner layer again. Okay, so a vesicle or a vacuole is just an inside-out cell, essentially. So let's pretend that's a protein. So my Golgi apparatus, here's my Golgi over here, just spit out a vesicle, and it travels to the cell membrane. It pops open and spits out its hormone, its protein, its lipid, whatever it is that the cell needs. Okay? So if we go back to the very first page, kind of puts it all into summary again. So there's two main types of transport, active and passive. Passive means no energy. Active means with energy. Simple diffusion goes right through the membrane. It doesn't need any help. We have facilitated diffusion, needs the help of a protein. And then we have osmosis, which is just diffusion of water. And then don't forget the three: hypertonic, isotonic, and hypotonic. Active transport requires energy. We have two kinds. Endo goes in, exo goes out. Penocytosis is drinking, and phagocytosis is eating. Okay, pretty simple. All right, guys, thank you much. And if you have any questions for me tomorrow, let me know. Bye-bye.